Today's general theme is going to be on model reuse and reproducibility. We're going to mostly focus on ModelDB and what we can get out of there, making sure that things that we get out of there work. And your homework exercises will be things like, hey, let's take something, let's adapt it, let's do something different with it. So that requires us to be able to run, and that requires us to be able to understand enough to change, right? Always make sure that you understand before you start going in and changing and messing around with things. Before we do that, however, I want to make sure, open the floor if there's any questions or comments about any of the parallel exercises that we've been talking about, right? Anything else that's come up. All right.
Well, good. Sounds like this group will be writing exemplary code. It'd be answering exactly the question that came up earlier this morning.
Excellent. Also, speaking of questions that came up earlier this morning, there was somebody who was having an issue with compilation of mod files. I'm not really sure what there's for that. I think when I switched my Mac to, what is it now? Big Sur, I guess. When I upgraded my Mac to Big Sur, I think it broke my Clang setup. And I think I may have had to uninstall Clang and then reinstall before compilation would work again. So.
Thanks, Robert. That was me. and it is a new install, so I was having problems with it. So I might have to go back and uninstall and reinstall.
Yeah, and when you do the reinstall, you don't necessarily need, by the way, you don't need all of Xcode. If you just type G++, it'll prompt you to install the command line tools, which is the only part that you actually need for NEURON. Whether or not you want Xcode is up to you.
No, actually putting Xcode in, because I did try that, it didn't help. It didn't solve the problem.
I don't know, but like I said, I think it broke mine when I upgraded to Big Sur, and I just had to uninstall and reinstall. And probably like having a partial mix is not great. I don't know. If that doesn't work, let us know and we'll try and figure that out.
Yeah, sure. I'll give it a try.
All right, thanks. Any other questions or concerns? All right, well, hearing none. So again, today's general topic is model reproducibility, reuse, probably maybe leaning a little bit more on the reuse side. But generally speaking, you don't want to start from scratch. There's a lot of work that's already been done out there in the community, and some of the best science is done by explicitly building upon that which has already been done. And one key resource for this is ModelDB. So ModelDB is a discovery tool and repository for computational neuroscience models. So the idea with ModelDB, for example, is you don't have to go and write mod files from scratch. You can go in ModelDB and you can find hundreds, or I think probably even thousands of them that have been parts of published models. And you can take these things, you can reuse and modify them. And we'll look more at that later. Obviously, once you get them, if you've got mod files, you're going to have to compile them within an IV modal before running them. And ModelDB will be kind enough to tell you, hey, look, this is a mod file that's been used in several cases, and you can go back and check those out and see, well, do you believe that each one of those, is that an appropriate domain, or are they all Alzheimer's research and you're not studying Alzheimer's, in which case maybe you want to think about it. And so the website for ModelDB is modeldb.yale.edu. And so we've got code, we've got metadata, we've got visualization platforms, and information that sets the modeling work in the context of other modeling work. So all of this citations is not with respect to global citations, like just because something's been cited by some paper doesn't mean it was cited for modeling context. So ModelDB tries to provide the sort of computational neuroscience citation graph. So what other modeling work has cited a paper? What modeling work has this paper cited? So it's a slight distinction, answers a different question that you might get from PubMed or Google Scholar. But fundamentally, the goal with all of this stuff, right? We want our code, we want everything that we make to be reproducible. We had a whole discussion last class, Ted led, about reproducible randomness, for example. We want to be able to make sure that even in the case where we're incorporating randomness into our models, we want to be able to give it to somebody else and they get the same results. Because, you know, and Karl Popper, the famed philosopher of science, points out, if you can't reproduce it, well, this is not a quote, but the quote is non-reproducible single occurrences are of no significance to science, right? If we can't reproduce it, then we're telling a story. But if everybody can reproduce something, then, you know, then that's actually much more useful. And then we can actually build upon that. We can count on it. We can analyze it. And it turns out it's really perhaps surprising for people coming into this field, but a lot of it's non-trivial to get computer code to just work on somebody else's system. Maybe Clang will decide that it won't compile mod files anymore. Maybe you know, but different platforms have different assumptions. One thing that I ran into maybe a year ago was that there was some disagreement about what a certain floating point number raised to the 4th power was. And the reason for that is that there are certain mathematical operations that that the IEEE standards have defined to be correct to within what's known as 1 machine epsilon, and other floating point operations like raising numbers to power, computing signs, and things like that are allowed to be off by what's known as 2 machine epsilon. And so this introduces the possibility for inconsistency. Well, okay, well, that's a problem. But we don't even usually have to get into these weird sort of edge cases. Usually if you've got this big complicated model, right, you're not going to put all of the complexity as best as you're going to try and describe it as well as you can in a paper. But there's going to be some detail of the complexity that is just not going to be in that paper description. And so one of perhaps the best ways to get somebody to be able to use your code and get the same results is if they have access to your code. And you can logically think of this in 2 parts, right? There is the model itself, right? So what is your system of interest? So you build up your cell class, you build up your network class, right? That is the model. And that's slightly distinct from the experiment, right? You know, that's not what you put in a neuron code. It's part of it. But the other part is the experimental protocol. So what are you doing to the model to produce the data? If you don't have both of these pieces of information, you're not going to be able to successfully reproduce somebody else's work. And I'm just going to skip ahead of my slides here because I really like this figure, even though it's a little old. So when people actually go and make computational neuroscience models, and we've looked at them in ModelDB, and we said, well, okay, well, how complex are these models? And so we have now, I think, something over 700 neuron models are in ModelDB, and I don't know, we'll see somewhere else, and a comparable number of non-neuron models. And so across these models, right, most of them, you know, more than half of these models are split up across more than 10 files. You know, a quarter of them have more than 20 files. More like a third of them have more than 20 files. Okay, so that's a lot of pieces of information. And these files can often be, you know, basically half of them are 2 kilobytes in size or larger. A quarter of them are 5 kilobytes in size or larger. There's a ton of information. There's a ton of complexity. And like I said, it's just really hard to describe this complexity in a paper. And even if you do, and you try to do this, any bugs, typos, errors, or omissions could completely change the dynamics. Although there are certainly tools that you can use. So like uncertain pi is going to help you detect whether or not you're sensitive to certain parameters or others. But there's a lot of complexity in one of these models. And again, really just sharing source code is the best way to get somebody to know exactly what it is that you've done. Out of curiosity here, has anybody actually tried to take a paper and just reproduce whatever's in the paper from scratch? Has anybody done that experiment?
Yes, I've.
And how'd that go? Well, I got a thumbs up. I don't know if that means it went well or...
It went terribly.
Yeah, it was an adventure.
Yeah, adventure is a good way to describe that.
So what
An
kind
adventure is somebody else having a hell of a rough time someplace far, far away. So for me, that sounds like an adventure. For you, it was not. Anyway.
So what kind of issues have people ran into when trying to reproduce work from scratch?
The problems I had was certain errors reproduced in code that was used across multiple papers, and just difficulty tracking down the corrected version. That'd be one example. So things like that. I also had to give a concrete example. I think I saw a mod file that had a particular error that allowed me to plot activation, like steady state activity.
I think I lost your audio partly.
Can think of that. I just couldn't pull the time constants out of the code. Was that somewhere in there in the code was messing with the model, but not necessarily in a way that wrecked the model, just in a way that made it difficult to examine to examine it more carefully.
So this was actually code that you got from someplace else you're talking about.
Yeah, it's a published model. I don't want to denigrate it because yeah, I had a lot of problems opening up certain mod files.
Well,
or not opening them up, using them.
Yeah, well, what about the problem of when you don't have the files, all you've got is the paper?
Well, yeah, that's a much worse situation.
Why?
Well, I guess you're right. Maybe it's not necessarily worse.
No, I mean, have you actually tried it? I mean, what problems did you run into?
So like I said, I was running a Hodgkin-Huxley model, but instead of just using the model as is, I pulled out the particular channel mechanisms. And in Python, I was just trying to see what are the taus. And it was impossible to plot that. And eventually what I did do is I retraced through a few different papers and rewrote the whole thing from scratch, and then finally was able to reproduce the activation curves and everything.
Well, you were very fortunate because at least you had some source code to work with, even if it wasn't doing what you wanted it to do. The major problems are that most method sections of most papers aren't complete enough for you to be able to reproduce the model. And most authors don't include all that information because they just forget it. I mean, it's been a while since they did the work and developed the code, and things that were at the top of their stack when they were developing the code have fallen off. They have a very top level view of what their model is and does and how it works, and they just don't remember all the intricacies. The reviewers don't want to see it. The publishers don't want to publish it. And since a lot of journals no longer accept voluminous supplementary materials, it gets kind of lost. And it's really hard to reproduce published work when you can't get the source code, even if the source code is busted. So you were able to succeed because you started, even if you started with inadequate source code, you were ultimately able to triumph. But most times you can't reproduce them.
Because the code was out there, I was able to compare a few different versions of the mod files and then detect the error and solve it that way.
Yeah, well, that's that's a different problem. That's embarrassment of riches. Too much source code, as opposed to none whatsoever. But except for very, very simple, straightforward models, it's very difficult to reproduce computational biological models from the papers that describe them. This is quite different from the kinds of models that are used in engineering and physics, where mathematical descriptions dominate. And once you've got the math, once you've got the equations, the conventions are widely accepted by everybody. Everybody knows the underlying theory, and there's never this kind of problem in the quote hard sciences. It just seems to afflict biology because we are so far from having a set of fundamental principles that everybody agrees upon. You know, and the few that we have are mostly drawn from physics and engineering and mathematics, and then we have to apply those to the biology. And when somebody says, I have a model of a hippocampal pyramidal cell, you don't know whether that's a single compartment with passive model, or if it's got branched tree, or if it's got realistic morphology, or what kind of channels are in it, or what they've bothered to represent in it. You have no idea until you get the code.
Yeah, so Natalie said something I don't know on the chat, and I don't know if she was thinking of what I thought, but do you want to say this out loud, because I have a specific comment on it?
It was essentially what has just been said, just that, yeah, like the little details and the both from the biologic standpoint and also from the simulation, like what are your time steps, what are your discretizations, like all of that kind of stuff is almost never included.
Yeah,
Yeah.
and that's like, and so it's not just even the model specification, it's not even just the simulation protocol, it's the experiment, but it's not just what's the experiment, what are the numerical details, because that might make a difference.
Yeah.
Also the units. When those are inconsistent, it's terrible to track down.
Well, and then there you don't know if they've actually converted them correctly within their code, or if they're using the numbers or what.
Yeah, a huge problem is the experimental protocol. And this affects not only computational neuroscience, but experimental neuroscience as well. When you actually use your model, how did you do it? You know, somebody says, well, we applied a such and such waveform with such and such statistics. And yeah, exactly, show me. And we changed the concentration of such and such at a particular time. Oftentimes, they don't even get to that level of detail in describing the experimental protocol. This happens also in wet labs. If you've done it, those of you who have worked in a wet lab environment and been to a few scientific meetings have probably come upon discussions at poster sessions or in the hall outside of a slideshow where you overhear, that was a beautiful paper. And then somebody else says, yeah, but you know, so-and-so's lab has been trying to reproduce that work for years and hasn't been successful. Can't figure out how to do it. And when we do it with our rats, we get completely different results. So it doesn't just afflict computational neuroscience, it affects experimental neuroscience as well. But we should be 100% reproducible. Everything we do is under our control and documented in the code, should be documented in the code. There should be nothing that we can't reproduce.
But here's the challenge though, right? So does having the code, I mean, how do people feel about this? Simply having the code does not mean that you understand the code. Simply,
or how to use the code. I mean, some of the worst models that I've seen look like beautiful code, but there's no instructions. Where's the main file? What do you do to change parameters in a simulation? How do you do that? I've got a file. I've got a source code here, which is about a little bit over a quarter of a megabyte long. Over 90% of which has nothing to do with the model specification. And I complain about spaghetti code that newbies write, but this is written by experts and it's well organized. But even so, key model specification bits are concealed in obscure structures and statements that you can't understand without learning lots about additional ancillary software that you're never going to use. There's never a clear, complete, compact, human readable specification of what's in this model. Sure, there's a gazillion functions and procedures that if you executed them might produce lists that contain parameter values, but there's no instructions for how to do it. And the only people who can reuse this code are the guys who wrote it in the particular lab that it came from. I don't think anybody's ever going to reuse that code. It's impenetrable.
And just out of curiosity, are you talking about the?
No, don't say it. I'm talking about what we talked about.
Yeah, well, so I'm just mentioning. So the other thing that's interesting is that you were looking at a 15-year-old code, I think.
15 years old, but it's human readable. I mean, I can read my way through. It's not badly structured in terms of structure. It's just that the comments are, there's no usage comments that give you any clue. You can't discover, you can see the cell classes, that you can see, and you can see the parameters that build those individual cells, but you cannot discover how the cells are connected. There's no place in there that has a single statement that creates a netcon and uses it to connect two cells or sets the parameters of that netcon. You can't discover the weights or the delays by looking at this source code. It's impenetrable. You have to, instead, you have to understand a library that is part of the source code, which is about 3,000 or 4,000 lines long and implements some kind of, I don't know, something or other that manages the network. And that's the only way you're ever going to use this, and it's just not worth the effort.
This is interesting. One of the things that is said in computer science, right? The person that most needs the documentation is you. The person who's most likely to go back and look at something is you. And so it can be you today, it can be you tomorrow. Can you of tomorrow understand what you of today were thinking? And if you're not providing enough documentation as you're writing to understand that, like, it's not just can you understand somebody else's, it's can you go back and understand what you did? Can you remember the assumptions that you made? Ted?
Yeah, it's an old saying that the only thing worse than wrestling with somebody else's old code is wrestling with your own old code. It's a very humbling experience. So
now there's a couple of
Today where I thought I forgot to include the, oh, sorry, am I audible?
yeah, we hear that.
For just a moment, I thought I forgot to include self dot on a single parameter. I almost had a heart attack because I thought all my results were junk. And then slowly comb through, and because I didn't comment it properly, I discovered that yes, everything was actually fine. But it is really annoying having to reinterpret your own thoughts or your own work if you don't show yourself what you did.
And so there are some things that you can learn to, there's some things that are just sort of good practice that you can learn. Like one thing that took me entirely too long to appreciate was don't use the same variable name for more than one thing, right? And that way you can make your code robust. You want your code, as you personally are writing, you want your code to fail if like that, you know, that if you omit the self dot, you want it to crash. You don't want it to keep running. Ideally, you know, to try to write code in ways that is going to let the computer catch your mistakes. But that's on the software design thing. There's some sort of philosophical questions on the chat. The last two questions. I don't know if you want to say them out loud. PG.
Oh, sorry. Hi. Thanks for responding back. I've seen this happen, like reproducibility and trouble with reproducing, not so much as in code, but also actually even with code, even with much simpler things, nothing complex as computational neuroscience. I've also seen this happen in experimental biology. I've also seen this happen in clinical research. So, okay, ModelDB, yes, I could release the code through ModelDB, but then that would be restricting it to just one place. But if I wanted to access somebody else's code and use it and try and reproduce what the other person has done, and if things, or based on what I'm hearing from you all, how would I circumvent the problems and make it work?
Well, one thing you can try to do is to contact whoever wrote the code. And there are people who actually do things right. Recently I ran across a couple of really nice papers written by somebody, did some extremely nice technical work and on the border between engineering and neuroscience. And the guy subsequently moved on to working at a different level of analysis and is now doing, I'm sure, excellent work in his more recent domain, which has more to do with behavioral level of analysis. But what he'd done was to make his code available through GitHub. He actually is a skilled programmer and knows the tools, probably has worked commercially for a bit before, or should say, at some point in his career, and so he's done it quite right. I think the prevention, an ounce of prevention for this is always better than trying to wrestle with cure. One does occasionally see attempts to reproduce published models. There is, what is it? It's, what is this? Remember what it's called, Robert? Which?
It's RE in parentheses science.
Rescience, right. And rescience isn't just limited, I don't think, to computational modeling, but it's also for anything. For example, if you're doing analyses of data, I mean, look, the people who do fMRI studies, for example, have grappled. I mean, there are three major problems, excuse me. Two are very technical, and they have to do simply with making the data available and also making available the algorithms that they used to analyze their data. And they've done a pretty good job of that. The third problem is designing experimental protocols that make sense and take into account the limitations involved in doing fMRI on cooperative or not cooperative subjects. And that's a serious challenge. But they're really trying to do their best to make the field honest, and they have adopted these policies uniformly so that it has become a standard within their domain. And they have meetings ongoing, not all the time, but every year or so, they have serious meetings about the latest sets of tools and how they're addressing the problem of modern availability and how they're accommodating the development of new techniques. We really need to do something like that in neuroscience, not just computational, but experimental as well. And we see some initiative from NIH and NSF in this regard with regards to sharing of and access to data. And INCF, for one, has made a big deal about things related to open science and what you might call the FAIR principles, which Robert, I'm sure, can address.
So I just want to point out, so very recently, I guess this all finished, got wrapped up last year. Frontiers ran a research topic on reproducibility and rigor in computational neuroscience. Disclaimer, I'm not an uninterested party. I was an editor for this topic. But yeah, so there's 16 articles in this research topic that you may be interested in checking out. The journal Rescience, they actually changed it recently to Rescience C. Historically, most of their stuff has actually been neuroscience focused, but it is not limited to neuroscience. You may want to check that out as well. And so these, okay, so this is, okay, two things. So there's this debate, and it's kind of silly, but what's the difference between reproducibility and replicability? And there's two different sort of conflicting definitions. So the research topic took made no choice and just said to please define by what you mean. But the two basic options that you have are either somebody takes a description in the paper and tries to run it from scratch, or somebody takes what you have and then tries to get exactly the same thing that you do. So if I make it from scratch, can I get qualitatively the same results? Does your result at least stand? Even if I don't get exactly the same, does your finding stand in this completely independent implementation? And in some senses, that's better, right? If you're working with a different set of code and it's all established independently, then there's two different ways of building something up, and you're finding the same result. That gives some hope that there's not, that the result that you're finding isn't a computational artifact, isn't a typo, isn't a bug, but maybe you are indicating something real. And so that's what rescience is trying to push. And I think it's a great thing. That is, however, not the scope of ModelDB, which is really like, let's go ahead and try to make your code, make it so that other people can use your code and build upon the exact code that you have. Now, you might say to yourself, but what about GitHub? Isn't that what GitHub is for? And that's true to some extent, and they both ModelDB and GitHub have, well, ModelDB links to GitHub now. It's just easier for the history. But ModelDB, being a domain-specific platform, allows us to annotate in a domain-specific standardized way, so that you can know, oh, hey, look, this is a model that has a transient sodium. This is a model that has an L-type ion, channel. This is a model that has an A current. So you can build all of these things up, and you can annotate them, and you can compare across other computational neuroscience models. We can build analysis and visualization tools. So this right here is what's known as ModelDB's version of ModelView. I think we saw it once briefly in NEURON, and as long as we have time as long as time permits, we'll go back to that later today as well, and use that to sort of look at a model from within NEURON to try and understand it. But yeah, so that's sort of the compelling use case, the main use case, the main advantage of ModelDB, being able to say, hey, look, who's done computational modeling on Alzheimer's or whatever your disease of interest is, or who's looked at A currents, or what are the A current models, being able to talk about it from a neuroscience context, being able to do visualization and analysis. And that's kind of the main goal of ModelDB. But certainly all of these other issues, it doesn't solve all the problems, but it does at least give us a place to start. So just to give you a sense as to what's in there, so these numbers are from a year ago, because I didn't update the slides, so sorry. But in any case, this gives you a sense as to what's in ModelDB. So 181 different cell types, representing from cells from across 21 or more species. We've got 58 different types of ion channels, pumps, and so forth, covering 169 topics. So this is the disease topics like Alzheimer's or Parkinson's, sort of conceptual concepts like spike time dependent plasticity, and more. Like I said, there's over 700 neuron models, over 500 network models. So there's a lot of stuff within ModelDB that you can use as starting points to build your own model, as examples to see what to do, how to use a function, to see what's been done in the literature as part of your literature survey. So, yeah. And we've already talked about this in our discussion sort of at length here. But fundamentally, as you're doing all of this, anytime that you find something, you should make sure that you understand it. And that's why I'm going to stress things like model view for looking at the structure of the cell. That's why we talked about, well, how does one change the synapse mechanism in something? And why it's important to have sort of clean codes that other people can go back, clean, well-documented codes that other people know where to go back and make changes and do them right. So when you're doing this, you know, remember that people may, you find some model on ModelDB or wherever, GitHub, you know, keep in mind that people solve their own problems. And so they may be solving a different problem than you. If they're looking at a disease model and you're not, if they're looking at a different animal species than you are, or they're looking at a different age of a certain animal, does it matter? Maybe. You should at the very least read the paper and figure out what it is that they're doing so that you can make an informed decision. Is it appropriate for what you're trying to study? Is their paper, you know, check, don't just look at one paper. Probably there's multiple models that look at similar topics. So if you find one paper, one model that's generating one result, well, is that consistent with what other models that are independent are finding? If so, again, that's cool and that's great. And if not, well, maybe you need to think about it and decide, really, really decide if you believe the biology that this is representing. NEURON provides tools. NEURON and ModelDB both provide tools for visualizing the model structure. Model view, obviously within NEURON. There's P-section as well within NEURON. You know, so then you may want to go and dive into these ion channels individually. Like, hey, look, this is a model of an A current. Well, who else? What other models have used this particular A current model? And ModelDB will tell you that. You can go in and look at their IV curves or whatever's of interest to you. And if you're not sure about something, the best way to get an insight into this is to make sure that you do have an experimentalist friend that you can ask questions to, that you can collaborate with. You don't want to be making up biology as you go along. So please, please try to make sure, and this is true for ModelDB, really this is true for any resource that you want to use. This is true for Stack Overflow, by the way. So people love to get programming advice from Stack Overflow, and that's great. But if you don't understand what it is that the answer, why the answer works, and you're adapting it for something else, that's also dangerous, right? So please, please, anytime that you find something online or otherwise, make sure that you try to understand it and don't just follow it blindly. All right, so obviously within ModelDB, this is not going to surprise you. At the top left of every page, we've got a search box. If you start typing in the search box, you start getting results. So you can do search for full text or attributes. So here I've typed 3 letters, HIN, and it's going to give me this like sort of structured results. So it's going to give me autocomplete suggestions, authors, cell types, regions, whatever, every sort of concept that matches this. You can, so the word completions here, you'll notice, are not necessarily English-based priority, but are instead based on priority within computational neuroscience as ModelDB knows it. So for example, the most likely thing that you're searching for if it's HIN is HINF because it's H infinity, right? So that's going to show up at the top of the search box. So it's going to try and give you these computational neuroscience related suggestions. All of this happens as you type, so it's real-time search results. You can also restrict to certain file types. So, you know, it's often very useful to, hey, look, well, okay, I'm trying to understand how to use this NEURON function. I want to find it within a Python file, or I want to find it within a mod file. So I'll search for a mod file that uses the word diffusion or something like that to see how to do that. And so forth. So let me actually just go ahead, instead of going through the slides, I do have a browser up. So let's just talk about, so let's just pull up an example, modeldb.yale.edu. I'll pull up model 87284. It's not at all random. This is so Ted is the second author on this model. It's senior author was my postdoc advisor. It's not at all random. However, so here's a model of amyloid beta effects on CA1 pyramidal cell. And what we see when we go to this model entry, so I found something of interest. Actually, let me just start from the beginning. Let's say that I'm starting out, I want to find about Alzheimer's. Hey, look, it says concept, aging Alzheimer's. Well, who's looked at aging in Alzheimer's? Not that many in the grand scheme of things. So here's 13 models that ModelDB knows about that involve Alzheimer's in some way, shape, or form. So I pick one that's of interest to me. And there's a little description here to make sure, you know, I think we all probably agree what Alzheimer's is, but there's at least in a high level sense, but there's a description for most of these pieces of metadata that describes what that actual thing is. So for example, if I click into the amyloid beta one that I was in before, well, what do I mean when I say INAT? Well, let's just click into it and it'll give me, you know, okay, well, this is a transient, a rapidly inactivating current. What's the deal with pathophysiology? You know, these are physiology-specific functions associated with a condition in which the normal functioning of animal is impaired. So for most of these entries, all of the metadata is defined. There are some exceptions where we'll find where the metadata is not defined. But if you run into those, please feel free to contact us with a suggested definition. But in any case, so we go to this model entry. It's every entry in ModelDB has an associated publication. So this is one way of providing a quality assessment. Somebody, somewhere, and it's not a bioarchive or an archive publication, although that is a persistent request. But somewhere, somehow, somebody's gone through a peer-reviewed publication of these entries with only a few historical exceptions. So we go in, we see, hey, look, this is a model that's associated with Alzheimer's, cool. It's got various metadata that's associated with it. So if I wanted to find other models that incorporate a transient sodium channel, I can just click on that transient sodium channel link. And here's a list of blah, 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 502 models that incorporate a transient sodium channel. We can go back. So here we've got a bunch of metadata. We try and get as much information as we can. Some of this is dependent, you know, we provide some metadata ourselves. Some of this is dependent upon whatever we get from the model authors who are sharing their code. So what kind of codes do we have here? So this is, I can click on model file to look at any individual, to look at the individual files. So here I've got the file tree. I can browse through. There's a bunch of mod files. The README is the default. If there's a README, that's the default file view. And so here, for example, the authors have included a couple of figures. So these relate directly to the figures in the publication. What we ask, which is enforced to varying degrees, but what we ask is that every ModelDB entry is able to generate at least one figure from the associated publication, or, you know, there has to be a clear relationship between the model entry and the figure. There are some models, and I'll tell you this, there are some models that provide simplifications. You remember that last time we talked about network models, and that we said that you might want to parameterize your network models so that you can run this giant model on a HPC system somewhere, but you could run a small model on your local system. And what you'll find sometimes is that you'll see, here's the code, it's set up to run on your local system, but you could change it to run a bigger model by, you know, changing the number of cells here or something like that. And if you're lucky, the model entry has nice little descriptions. This is a decent set of descriptions within the README file. Always a good place to start. That's why it gets the name. You can go, you can click in, you can say, hey, look, well, look, cagk.mod. What's the calcium activated potassium channel code? Well, I can click on it, I can look at it. I can see the parameters that are hard coded into the mod file. Question for you. If I see that let's say GKBAR is 0.01 per ohm per square centimeter. If I see that conductance, does that mean that's the conductance used in the actual simulation?
Depends on the rest of the code.
Yeah. Right. So it could very well be the case that somewhere there's a line in Python, or in this case, Oak, that goes back and says, you know what, I want you to insert this ion channel, but also change the conductance. So this is one of the sort of gotchas of looking at code, right? Even if you have the source code, being able to understand it from the perspective of source code is non-trivial. Because how do you know that you found that last line that changes GKBAR? You know, maybe it gets changed in several places, and what is done in the last order? It's tricky. And so what both NEURON and ModelDB's solution is to actually go ahead, try to run these try to actually run the simulation, and then just ask the simulator, hey, by the way, what was the value for GKBAR? All right, and that's the secret behind ModelView. We actually go, we run the simulations, and then we extract the parameters from the simulator, and we don't look at the source code at all directly. And we'll get to that in just a second. The one other thing that I want to point out here, well, let me point out a couple of things while I'm here. You'll notice that there's this fancy little asterisk next to the file name, cagk.mod star. What's that star about? Well, if I mouse over it, it says click to see other models using this file. Well, that's interesting. Let's do that. And so here I get a list of, what is it, 1, 2, 3, 9 total different other models that use the same exact representation of a calcium activated potassium channel. And I see that this particular calcium activated potassium channel has been used in a CA1 pyramidal cell, and it's been used in a CA3 pyramidal cell. I see that it's been used in a very recent model involving schizophrenia. So it's been used in a model involving schizophrenia. It's been used in a model involving Alzheimer's. And so you can go and you can look at the different contexts in which these channels have been used, and that can give you some sort of sense, and that can help you decide if you believe that this is right for your usage. And so if I were to click in any one of these, this is exactly the same file. And so right now, that is the rule. We are not currently assessing for equivalent files. It is the text of the file, we pay no attention to the file name, is the text of the file identical? But so we do look for duplicate files in that way. One other thing that I want to point out here while we're looking at this, anytime that I'm on a mod file, well, this isn't quite true. Many times when I'm on a mod file, I'll have this IC genealogy link, which is for the ion channel genealogy. So I can click on the ion channel genealogy, and so these are friends at Tim Vogel's lab, formerly at Oxford, hence the URL, currently in Austria somewhere, I believe. And so they've gone through a large number of ModelDB entries, specifically NEURON entries, look through the mod files, and have assigned additional metadata to the mod files specifically. So this is going to tell us that historically, that representation of a calcium activated potassium channel, well, it goes all the way back to this thing from 1983, which pertained to data from a rat, the CA1 cell. So, okay, so it really makes sense in the CA1 context. What about the CA3 context? But so then they go and they have all of this additional information, this additional metadata. They've ran a series of analysis tools, and so they provide this graphical channel browser here that you can pop up. And so there's you can see the relationship. So what other calcium gated potassium channels? So so there's the one that was in ModelDB 101.629 is related, but these ones in different clusters either have different kinetics or are otherwise known to be differently related. So this is a clustering algorithm based on citation family, I believe, but you can also look on similarity and channel similarity. And so they provide these kinetics. And so you can get a sense as to different sort of clusters and different representations and different ways that people have found. Yeah, Ted.
Oh, did I accidentally? Oh, it turns out that he is at IST, which is an excellent location, and should get a lot done there. It's just about 7 miles north of Vienna. Excellent science going on at IST.
Excellent. So I also want to point out, so I've been kept, I keep talking about model view. So let's look at model view from within ModelDB. So there's a model views tab on many, but not all of the NEURON models. It's most likely to be there if it's an older model. If you have something that you think we should have a model view for that we don't, just let us know. You can go under load model views and select a particular figure. And this is essential because it could very well be the case. And you'll see in your homework that where the different figures are, you'll see in your homework an example where the different figures represent very distinct models, at least in the sense of, well, the cell morphology is different. So anyway, so here's what you get. So you click on model views, you load a particular model view. Let's say that I click inside this one cell with morphology. Well, okay, there's one cell, the root node is called soma. Cool. In total, that cell has 194 sections and it was run with 974 segments. You could imagine that you could rediscretize this and do this differently. You can look at the ion channels and other mechanisms that are inserted under however many inserted mechanisms. So if I click there, well, obviously we're always going to have axial resistivity and we're always going to have membrane capacitance. But what else is going on? Well, there's a whole bunch of ion channel mechanisms that are inserted, and there are links to the various associated mod files. And you'll see sometimes the mod file name, sometimes people follow convention where the name of the channel matches the name of the mod file, but they don't always. So this like KAD versus KADist, right? So the mod file is called KADist, and I can click on that, and this is the mod file that's associated with it, right? And the name, however, is just KAD, the suffix in the mod file. But in any case, let's look at KAD just for kicks. So if I click into there, I can see the parameters that are involved. So here's GKBAR. And so if I were to look at KADist, where is GKBAR? There you go. GKBAR is 0.008. That's it. It's one number. Except when I look at the model view entry, I see that GKBAR is actually almost never that, but is instead something much higher, and it's distributed in a way that seems to roughly increase with distance from the soma. So I can tell at a glance what is actually used in those simulations, as opposed to trying to track down, well, okay, well, where is this defined? Wait, has it been changed since then before the simulation? So the way this does is it runs the model until the first time step, until the first F advance, basically, which is a single time step advance. We've been using continue run to run for many time steps, but until the very first time step, and then it says, well, okay, well, NEURON, what is in your model? And the assumption there, which is not 100% true, but is almost true, the assumption there is by the time of that first time step, that model has been set up. And we provide various ways of handling, like, if there's a clear initialization phase, we do skip that. And there are ways that we are able to address that. So in any case, so this lets us see at a glance, well, okay, here's the deal with Ka dist. What about HD? How's that distributed? Well, I can click into there. Oh, well, once again, that increases with distance from the soma, but that literally increases with distance from the soma. It's a much simpler relationship than whatever we see when we look at the distribution of A-type potassium conductances. And I can look at this and I can say, well, okay, well, it looks like it's brightening up on these obliques. Let me move out to the obliques. You can see that on the right graph. Well, actually, let me do this. If I move along the space in the top here, you'll see I'm hovering over the right graph, and I'm at one of the local sort of peaks of one of those lines. What's also circled in the middle graph, it's showing, hey, look, that is at the end of an oblique end right here. So I can sort of get the sense that once I go into the obliques, it gets it increases at a faster rate. But I can see all of this, and good luck trying to find that out from looking at the code.
Hi, Robert. Is this when at the resting state, the value is changing throughout this model, or?
So this is the sorry, the bar is indicating the peak conductance. So if you look at the, this is not like the gated conductance. This is, so we take GKA is GKBAR times the gating variables. Does that answer your question? Right. No.
Yeah, kind of.
But that's another reason why we link back to the. So I guess the most direct answer is that in this particular model, GKBAR does not change over time. But generally speaking, the NEURON convention, so if you look at like the hh.mod file, you'll see these GNA bar variables, which are the peak conductance. But that's just a convention, and never trust conventions. Never depend on conventions. Follow them, but don't depend on them.
Well, actually, the GKBAR notation is really meant to be, this is the channel density. Okay, this tells you how abundant the channels are over the surface membrane. It doesn't tell you that that's the peak conductance, because that conductance level may never actually be achieved in the course of your computational experiment or in the course of the animal's life. It would only be achieved if all of the ion channels were open at the same time, and that rarely occurs. So GKBAR is simply a statement of the product of how many channels there are per unit area times the unit conductance of each channel.
Oh, I got it. Thanks. Yes.
You're welcome.
Yeah, so the units are reciprocal ohms, reciprocal area. And I think we saw in the last discussion about how that you can actually query NEURON to find out the units of a specific variable. All right, well, I've seen that A-type conductance is interesting. H-type channels are interesting. What about some of these other ones? If I look at this T-type calcium, nope, that's pretty constant. That's constant everywhere. And I can see that at a glance. I know that I don't have to go diving into the code. And we'll see that we'll see that NEURON provides a similar introspection capability. Just to throw out some of these other tabs that are on here that might be interesting to you. The citations tab really, like I said earlier, tries to put everything in the context of the rest of the computational neuroscience literature. So we're not trying to keep track of all publications ever, and things get cited for a variety of reasons. But for example, we know when we, here are the following things that we know cite this particular paper. You know, so we've got so this was a 2010 paper. There was a 2012 paper that came out of Michele Maiori's group that cited this. And by the way, here's the computational model. You can click on that, and then you can look at that ModelDB entry. And so anytime that there is a ModelDB entry that cites something, or this cites a ModelDB entry, that's going to appear in bold, and then you can click on that, and you can look at the other one and see, and you can get a sense as to how these model entries relate to each other. To throw this out there, there's also this simulation platform link. If I click the simulation platform link, what this does, and this is only available for some of these things, but for those that it is available to, it gives you this option of launching a virtual machine wherein you can run NEURON remotely through the cloud. There we go, it's starting up. It's connected to Guacamole. There we go. So here we have, I've downloaded, now you're in the awkward environment of watching me screen share as a virtual cloud thing. But nonetheless, so here we go, we get this access to the model. I don't have to download, I don't have to install anything, I don't even have to explicitly compile because it's done that before, but I just click that button that says figure 1, 2, and it's ran that simulation, it's displayed that. I could click the button for figure 3, and I guess that's it. That seems underwhelming. But that was interesting. So here's figure 5 button with running a whole bunch of simulations. You calculate the peak back propagating action potential over time, etc. So this lets us go, this lets us run cloud simulations without having to install NEURON. I wouldn't necessarily recommend this for any serious research. It does have a 5-minute timeout, which is what that ticking thing is. But it is a way of quickly exploring a model, especially if you're on somebody else's computer and you want to be able to demo something.
You know, what this really illustrates, though, is ModelDB's interoperability with other online resources. That simulation platform thing is developed and maintained by somebody else at a different institution, and our resource, ModelDB, and their resource, simulation platform, are able to interact transparently for you. So whether or not it's actually useful in any particular case depends on the model, but it sure allows you to do things without actually having NEURON running on your local machine. The nice things about what Robert has been showing you previously, I like the model view aspects particularly, and those fall out of the fact that NEURON has such powerful introspection capabilities, so that this looks extremely complicated and difficult to set up. Well, it required a lot of development to come up with the original idea and bring it to its present status of functionality, but a lot of this stuff that's being shown is discoverable as a consequence automatically of NEURON's introspection abilities. And other simulation software could conceivably have such a feature, and it could be implemented and added to ModelDB, but nobody is, most of these other developers are not bothering with this. They're not working on their own model view tools. Maybe they feel that their code is sufficiently clear and everybody who's using it is sufficiently smart. They're not going to have any problems. Maybe they have smaller user bases and they don't worry about, you know, anybody that they don't know trying their code because they're happy with what they're doing and they're being perfectly productive with the jobs they already have. but there is no reason in principle why other tools could not have similar model introspection capabilities. And we'd sure like to incorporate them in ModelDB if they did add that. I think one of the more valuable things here is the fact you can actually see what the simulation protocols are at the very bottom of this list. Under references, you get these run protocols, and this is actually, how do I use the software? And that is often extremely difficult to discover when you get a bag of software from any source. Somebody says, oh yeah, I'll let you have my model of the human brain, and then they give you 10 terabytes of code and associated data and a complicated directory architecture. And where do you start?
That's very true. So not all neuron models have a model view that's available, but when they do, all of this information is available in a structured form. And fundamentally, it had to be available in it had to exist in a structured form, because otherwise we wouldn't be able to generate the model view. We had to be able to have a clear way of, this is how we run it, this is how we compile the mod files, this is how we run it, this is how we launch this particular simulation.
Excuse me, when Ted told other tools don't have a ModelDB, which tool he was intending? Like, for example, like MATLAB or something? Or
so MATLAB's, so once you go into these sort of general purpose programming languages, they provide introspection, but it's hard to map that back into understanding what it means in a biological context.
Yeah, the first thing you lose with MATLAB is any notion of sections. There's everything is compartments, and you have to remember how many compartments are in the model and which compartment number corresponds to which part of your cell. There's no, it's great. It's great software. It's extremely powerful, and you can do a lot with it. And in fact, if you look at ModelDB's contents, the most numerous models in ModelDB are, of course, neuron models, because we follow the literature for neuron models as part of our grant maintenance. We have to keep track of how much usage there is. So we know, and we try to solicit as many as we can and try to get as much of them in as possible. MATLAB is the second most popular simulation tool represented in ModelDB, because there's so many MATLAB users making neuroscience, computational neuroscience models with it. And right now it's more than half, the number of MATLAB model entries is more than half the number of neuron model entries. So between these two, they account for a huge fraction of what's in ModelDB. And we're extremely interested. I mean, oftentimes people using MATLAB want to use Neuron for certain things, want to combine MATLAB and Neuron. And there's often Neuron users who want to do things with MATLAB. Sometimes it's data analysis. Sometimes they want to take advantage of a MATLAB model without having to re-implement it in Neuron with either Python or Hoke. And we're very interested in seeing this kind of thing happen. And who knows, maybe that can happen in the near future. We'll see. Did anybody here use MATLAB a lot? Can you raise your hands if you do? Okay, thank you. Do you find yourself on occasion wanting to do things that involve that have a workflow that involves Neuron and MATLAB?
I just work through Python, but like MATLAB into Python into Neuron,
Okay.
which is it's not pretty, but it works.
Yeah, yeah. Well, it's cool that you can actually do that and get it to work. It would be nicer if it was more transparent, but at least you can get that to work because of Python. So.
So just to give you an example of here's a model entry, so where the compiling is not always straightforward. So sometimes, yikes, that one is extremely not straightforward. I think there are easier ways to do that. But in any case, this is a way that we know works, and we know it has to work because we tested, because otherwise we wouldn't have generated the model view. But so sometimes the instructions are non-trivial. And so that's why if you do have problems and there is a model view, go ahead and look at the instructions and see how to compile them if there's anything special. Right? Those run protocols are always in the same standardized 3-part thing. They're always at the same place. They're always at the very bottom of the model view. The one other thing that I do want to highlight, just because, well, okay, so this one, for example, is a model that ran at 34 degrees Celsius, but didn't include any temperature-dependent ion channels. If I go back to the Alzheimer's model, this model was run at 35 degrees Celsius and included 10 temperature-dependent ion channels. So just remember, I guess I just want to remind you that temperature matters sometimes for some of these models. And so we do try to provide that little hint for you when you're looking at this stuff. Also, fun fact, some of these models have this 3D print tab. So ModelDB is part of what's known as the SenseLab suite of databases, which has a number of databases of varying utility. So this one is the 3D ModelDB, which has a bunch of 3D printable models that you can go and download. And so then this is a picture of a 3D printed pyramidal cell. Specifically, this is that 3D printed pyramidal cell of the model that was in that Alzheimer's model. Printed in Scharfenberger dark chocolate. This was printed with so this was printed with Shapeway's selective laser sintering is how this one was printed. At the time it was called Black Strong and Flexible Plastic. I think they've changed the name though. But they have a wide variety of stuff and it's all relatively inexpensive to go ahead and print. Also within SenseLab, just to give this a shout out, so we're looking at, for example, we see all of this metadata down here at the bottom, search NeuronDB for information about, well, you know what, I'd like to know more about CA1 pyramidal cells. I can click on the link and that will take me to a thing called NeuronDB, and I can just go to the overview tab of CA1 pyramidal cells. I've got links to other resources that I might be interested in. In particular, we've already talked about neuromorpho.org, which has morphologies and other reconstructions of this. I could go to neuroelectro.org for electrophysiology properties. Well, I'll pull it out and then we'll come back to it when it's ready. If I go to neuroelectro for electrophysiology properties, I could look elsewhere for other properties. I can go to this, let's say, plus connectivity tab, and this will tell me more about what's known biologically. So I know that in the distal apical dendrite, there's A currents, there's T currents, H currents, and so forth. We have some information about what's in these various sections. If you'd like to know why we know that, just hit the plus classical references notes, and that'll give you the same view, but much more expanded with whatever sort of data that's known about this. These are the classical references because much of this information, this level information, has been known for a while now. So we're not telling you everything that mentions an acorn in the distal apical dendrite, but we are telling you some references that talk about the acorns, the distal apical dendrites of these cells. And there are other things that you can do with NeuronDB, but the big thing there is what cell types and what ion channel types are known to be present in what regions of the cell. And like I said, from the overview links tab, you can go somewhere else. Oh, here we go, NeuroElectro loaded. So NeuroElectro is a resource of electrophysiology properties. And so what they've done is they've analyzed, does it tell us how many? Looks like 122 entries that have mentioned a hippocampus, have measured something about a hippocampus CA1 pyramidal cell that's of electrophysiological reporting. So for example, the input resistance, well, the measurements have varied wildly, and you'd have to, but you can mouse over each one of these dots and you can see where any individual measurement comes from. Right, so here, this is a model, this is a paper, this is a real cell, that actually existed and was alive at one point. But in this particular cell, this measurement that they got was in the case of a certain gene deficiency. Right, so maybe this looks reasonable. But so they'll tell you all of the measurements that they know about, and you can go in and you can follow through their literature, try to get a sense of it. Here's the mean and standard deviation of all the known measurements. And you can see that this input resistance of the CA1 pyramidal cell is somewhat lower on average than the input resistance that they found across the literature for all the neurons that they know about. And so that's input resistance. They have resting membrane potential, lots of electrophysiological measurements, not all of which are obviously available for all cells, but what they have available on their website. And of course, there's an API, and you can go and query this. And you can use this though. So, hey, I'm building a model of a CA1 pyramidal cell. Is my spike half width reasonable? Well, if your spike half width is 5 milliseconds, then no, it's well outside of what's been seen in the literature. So you can sort of use this as a basis point. And there's things called like NeuroUnit that can help you validate your models against various sort of properties to see if, they're matching whatever kinds of measurements that you want to reproduce. And we've already seen EFEL from the Blue Brain Project, and there's Elephant is another library that's used for measuring sort of electrophysiology properties. So in any case, there's this. So let's see. Let's go back to the slides because we've just been kind of talking. Yeah. Okay. Model view is cool. Oh, if I'm in this 18 inserted mechanisms, I can mouse over it and I can see, hey, look, in this particular cell, well, what's that section called? Actually, what's that segment called? Where is it in 3 space? What ion channel mechanisms are present? We've already seen, we can see these heat maps. We can see the references. We can see what's been reused. This is another view of that little asterisk information. Hey, look, this this calcium-gated potassium channel's been reused in these other contexts. You can check them out. We've talked about ion channel genealogy, the metadata that it provides. And you can,
Robert,
yeah, go ahead.
this information is provided by the people who upload the model.
You talking about the metadata or?
Yeah, metadata, yeah.
So yes, so if you're talking, let me go back.
Yeah, here, these things.
Well, this is all automatically derived. Everything in model view is automatically derived.
Oh, wow.
From an analysis of either the code itself or across ModelDB. So for example, it knows that these mechanisms are there because it asked NEURON, because NEURON tried to run it, and that's what NEURON said was there. It knows that this mechanism depends on the potassium NERTS potential because it analyzed this. So no human had to manually go and curate that. That's all automatically derived. But the metadata that you see here, for example,
right, these ones,
that was entered by a human at some point. Although we do have tools to assist this.
Okay.
And so that human, ideally, the person who knows the most about the model is whoever the model author is. And so ideally, the model author is the one who's telling us what ion channels are there, what cells are there. If they don't, we will try to look at the paper and find out this information as best as we can as part of the curation process. But it's much better if we get it from the expert, which is the one who made the model.
Okay.
So I just want to point out that once you have this resource where everything is aggregated, like I said, there's 700 and some odd 50, I don't know, neuron models, right? Then you can start asking questions in these meta literature reviews like, okay, well, what ion channels have been represented in CA1 pyramidal cells? Well, there's 47 models with CA1 pyramidal cells and only CA1 pyramidal cells. So we don't have to worry about these multi-cell things. But there's 47 models that have just CA1 pyramidal cells that include an A current. Maybe an A current is pretty important. And there's a total of 26 different channels, currents that you can find if you look across ModelDB that have been mentioned and that are included with a CA1 pyramidal cell. And so this gives you a sense of sort of a meta literature review, lets you see what other modelers have included. Because just knowing that something is there doesn't necessarily, if you know when you're doing a model, right? You're by definition trying to simplify. You know, you're trying to build something that that reproduces the key aspects that you're interested in. You're not trying to include every detail that you know. But 47 authors have thought an A current was pretty important, was important enough to include. Cool. If one of those currents, one of those 26 currents that have been included was only included by one person, maybe you don't need it. Maybe that's not a baseline thing that you should understand. Yeah, maybe it's there, but maybe it's not actually relevant to a lot of kinetics. Now, if you're studying the same problem that that person's studying, you know, then maybe it is relevant to that particular problem. So this gives you a sense, but it's only a sense. You have to actually read the literature as to what is sort of generally important versus what might be relevant to a specific problem. And so there's lots of ways to use ModelDB. So obviously we've talked about this, like, hey, look, I found a model. I want to download it. I want to experiment. I want to understand what the model's predictions are. We've seen using model view to try and get a better sense of the model structure. You can also go back and just try to look. We did that search for Alzheimer's. We saw, well, okay, what are all of the Alzheimer's models that we know about? So we may want to look for Alzheimer's models or a specific type of model components, like what are all the L-type calcium channel models? And if I swap them out, do I get the same results or do I get different results? And the other thing that people use ModelDB, or another thing that people use ModelDB for, is just using it as a source of examples, right? With over 1,500, I think over 1,600 ModelDB entries, you know, this gives you a sense of, hey, look, for whatever simulator is your favorite, I want to know how something works in NEURON or MATLAB or whatever. Well, let me go and look at all of these examples that I can find in ModelDB. And those are examples. Somebody somewhere has used it. Now, you're going to have to decide, again, don't just parrot whatever you see, but decide if it makes sense. But it does give you some examples as a starting point for basically, you know, across pretty much any simulator you want, how a certain term might be used, how a certain simulator tool or even language feature might be used. And that can help you as you're building models. The flip side of this, of course, is sharing models. We talked about earlier, but I do want to reemphasize this, not directly sharing models, but when you're building the models, try to write things in a way that's as clear as possible so that you can share with, write it in a way that your advisor can understand, write in a way that, you know, your lab mates can understand, write in a way that tomorrow you can understand. You know, if you have documentation, if you have clear modular components, you're going to make it easier for you and for others to build on your work, and you're going to make it easier to believe in what you've done and to believe that you've done what you think you've done. And as you're building these things, you know, use NEURON's model view and go back and check and see if you do get the same results. See if, you know, if you look at model view in NEURON, and we'll do this with that one Alzheimer's model in a little bit, but if you go back and you look at the structure that NEURON says it's using, and it's not what you think it's using, well, first of all, NEURON knows what it's using. You should probably check that. And if there is a discrepancy between what you think is happening in your model and what NEURON thinks is happening, you should definitely track it down. All right, but so you want to build things, you want to design things with the purpose of being useful both to future you and possibly to other people. And if it's useful to other people, this is good for you because then they're citing your paper, right? And so there's a little bit of self-interest here as well. Okay, so how do we do this with ModelDB? So, you know, if I want to share with ModelDB, I can start out, I can hit this big blue submit. I go to ModelDB's homepage, looks something like this. I can hit this big blue submit model button. Once I hit the big blue button, I get a form. And the actual required part of the form is fairly brief. It's who are you? How do we get a hold of you? Give us a zip file with your code, because you can't share the model code without sharing the model code. Give it a, so we don't require, ModelDB has the ability to do logins, but this is really not necessary unless you're doing administrative stuff. So what we actually ask instead is that you use what's known as a read-write access code. So basically a per model specific password that you can share with somebody else. You know, you can share with your lab mates if you want to make a change. It's not your account personally. Nobody has access to your stuff, but this is one specific model that your lab has been working on. You can update collectively. So you can assign an access code for making changes. And if you wanted to make it public, we do, as we've mentioned, require a citation. It has to have gone through peer review. So just go ahead and throw in a PubMed ID citation, whatever format. This is actually entered into the database in a structured way through a semi-manual process. So we have some flexibility. You can put it in whatever format's convenient. Now you could just hit that little itty bitty blue submit button here, because this gives us the information, right? We know who you are, we know who made it, we have the code, we know what publication. But we make this little button small because we don't really want you clicking that just then. What we want to know, we've talked about this, we want to know all of this metadata. And down below, you see where it says additional information. Down below, there's a whole form that you can fill out. But we gave you another big blue button. And if you see a button, you press the button, right? That's what buttons are for. So you press this big blue button. And it'll pop with a form that says, hey, why don't you go ahead and paste your paper abstract here? And you don't have to paste the abstract. You could paste a description of the model. You could write any extra words that you want if you want to sort of describe what the model is. And this doesn't go, this gets forgotten about basically. But what we do is we apply a rule-based system to whatever text you input here. And it comes back and it says, hey, it looks like, based on our understanding of what these words mean, it looks like that you're talking about the following things. And so this is what our system identifies as being a thing that your model is about, your paper is about. These are the keywords. We've mapped we've mapped the words in your paper to our metadata, and these are what we think the model's about. Now, fun fact here, you'll notice that cell type didn't show up for this model, right? And that's because there was no discussion about, in the abstract, about what cell types they're looking at. And so this could go one of two ways, right? Maybe the cell type, you know, isn't that important to the abstract. Maybe it's not the story. And in which case you probably don't want it to appear in the abstract, but you do want it to appear in ModelDB. So you could just go ahead and say, oh yeah, and we like CA1 pyramidal cells too, or something at the bottom of this box, and then it'll add it. Or secondly, as you're doing this, and if you're doing this before you've actually submitted the model, before you've actually submitted the journal, you know, if it comes back and it's missing something that you think is key to your story, like if that CA1 pyramidal cell fact was actually very explicitly about the story, and I'm not saying it is here, but if that was part of your story and it didn't show up in the abstract, if it's if whatever's important doesn't show up when you do this automatic identifier, maybe go back and reread your abstract to see if it showed up in the abstract. And maybe it did, and the identifier just missed it. And if so, shoot us a line, tell us what we missed, and we'll try to improve the identifier. But if not, that's a chance for you to go back and actually make sure that your abstract is hitting on all the things that you think are important. So another reason to go ahead and submit to ModelDB prior. So my suggestion for you would be to go ahead and submit to ModelDB prior to publication, prior to submitting to the journal. Go ahead and then put the ModelDB link in your journal publication, you know, so that people know what it is. Now it's not going to be public, but what you can do is you can send a note to your editor saying, by the way, the read access password is this. You know, here's a password that people can go and look at it. And then that way your reviewers can go and look at your code, because sometimes they're going to get cranky if they don't see it, if they can't see your code. It really depends on what reviewer you get.
Yeah, in practice, in most journals, there's nobody going to sit down and work through your code and make sure that it's beautiful and totally Pythonic or any of that stuff. Just the fact that you made the code available signals to them a certain amount of confidence that you have in your work, and they're liable to say, okay, we'll suspend our total skepticism about models since this person is willing to put their code on the line. Now, there may be somebody there who really has serious doubts about what you did, and if they can actually start your simulation and see the results for themselves, that may be very helpful in influencing them toward acceptance. Another thing is that some reviewers will say, we want to see that you're making your code available. And so they want to actually see in the manuscript a statement of code availability that maybe actually includes an explicit URL. And if you've already submitted to ModelDB and the code is there, we'll keep it secret until you tell us that it has been accepted for review, accepted for publication. And once that has happened, we can release it. But if you submit the manuscript and it's got the link in there, and they know that you've done that, and they can use your, you can get a private secret code for them that gives them the ability to just look at it. Again, that may give them some more confidence in the acceptability of your work.
Yeah, it's a good point. And again, not all reviewers will care, but some reviewers might. So yeah, so obviously when you do this, we do ask that you actually check these to see if they make sense. And so the way that we do this is you can deselect anything that doesn't make sense, and then just hit, once you're happy, whatever's selected, just go ahead and submit. Click this accept selected keywords, and then it's going to populate this structured form. And there's a lot of stuff here, but it's going to populate the structured form. Here are the model currents, here are the model concepts, and so forth. And as you scroll through this structured form, you can say, oh, you know, hey, look, there's an A current, but you know what else? There was an H current, and there was a sodium current, and, you know, whatever. So as you scroll through this, hopefully this will hint to you other things that you might want to to mention, and you can start typing in these fields and they'll autocomplete. And they'll show you autocomplete suggestions. Thanks. And these other fields are sort of just generic. You can go ahead. If you can't find whatever you're looking for in the structured data as a pre-existing ontological concept, then you can go ahead and free type it, and then we'll try to map it to some concept that maybe links either to what we already have or to a new concept. Oh, the one other thing that I meant to say earlier, when you're submitting this, and especially for these, with journals more and more, Ted mentioned the FAIR principles earlier. So FAIR principles are the data should be findable, accessible, interoperable, and reusable. One of the key aspects of reuse is actually having a clear license specification. So we ask, do not at present enforce, but we ask that you, when you're submitting one of this, go ahead and talk with your PI, think this through. What license do you want to release the code in? Who is allowed to use it? And so commonly these, scientific things tend to get released under an open source license. So BSD, GPL are fairly common options. There are certain restrictions that GPL has that BSD doesn't. MIT is an extremely permissive license. But yeah, by explicitly saying, you can use this code, here's the rules, I'm releasing this as open source, then you're making it so that other people know that they can safely reuse your code. Right? You're establishing a ground rule, because otherwise it's not fair data sharing. One thing that ModelDB does insist on, and I'll show you this actually, if we go to ModelDB and I look at any page of this, down at the bottom of every page is this how to cite. And so our instructions for citing ModelDB, if you're using ModelDB, so obviously if you're using ModelDB to provide models, please give the URL, and we ask that you cite this specific paper. Came up before. This is a 2017 paper. If you're using a model from ModelDB, then we ask that you cite both ModelDB and the paper that described the model, whatever the original model is. So those terms are ModelDB's terms of use. You have to cite us if you're using something you got from us, and you have to cite the paper that you're using it from. But it's possible the individual model may have specific rules that go beyond this. And I think the most common one that you'll see is there are certain universities that don't like open source licenses, but like to release things with non-commercial licenses. If you're a commercial entity, obviously you have to respect that. If you're doing an academic thing, then that's fine. That works for you. All right. One other thing, so we are working on updating ModelDB's look and feel. So slightly more streamlined, to put the analysis more up front, to make it easier to do those sort of meta-analyses. Well, what are the most common ion channels in an A current and stuff like that? Cleaning up the API. Both ModelDB has an existing API, but if you go to modeldb.science, there's which is our development version at this point, there's a cleaner API. But some of the data, it hasn't been synced lately. So we're working on that. We're working on updating this. We're certainly happy to have any pull requests or GitHub issues that you think would make this a better resource. So yeah, so that's where we are. That's where we're going with ModelDB. I do hope that you consider sharing your code, and I do hope that you look when you're trying to build something, when you're trying to get something started, to see what's been done before, and if that's a framework that you can use, and if not, to really think about why not and how to make sure that your own work is reusable. Just to touch on a couple of other things that exist. We've already talked about neuromorpho.org in the class. For those of you watching on YouTube, I'll add a link to that discussion in the description below. But from Neuromorpho, we can go and we can get access to over 100,000 neuron morphologies. You can download these, you can run them, import them into ModelDB, import them into NEURON, run simulations, or add ion channels and run simulations on them. And they provide a massive amount of metadata as well, and you can decide if this is appropriate and consistent for you. We've already said, and this is true for ModelDB, this is true for really any resource, right? Whatever you see, you can't assume that it's made for you. You gotta look at the paper, you gotta see, is this relevant for my, my actual use case? We've already talked about with the Neuromorpho some specific qualitative tests. I just wanted to throw them up there on the slides in case you'd forgotten. Again, the longer discussion will be linked in the description on YouTube. We've seen NeuroElectro. NeuroElectro is a resource of these electrophysiology properties that have been measured. You can compare cells to each other by property. You can look at an individual cell and find the various properties. It doesn't make any attempt to explicitly distinguish why the values are different. That's up to you, but you can at least see the range of values that have been reported. And one other resource that I want to give a shout out to is Open Source Brain, which is basically a platform for collaborative development of computational neuroscience models. Most of the models that are in there are being developed using a data exchange language called NeuroML. And there's a tool called JNeuroML that will convert NeuroML models to run in NEURON. So anything you find on Open Source Brain should be convertible to run with NEURON using JNeuroML. And like I said, it's a platform for collaborative development. And finally, I just want to point out, well, not finally. So pretty much all of these resources have Twitter pages. You can follow them to see what's going on, to see what's new. So SenseLab, which includes ModelDB, is at SenseLab Project. Open Source Brain, Neuromorpho, Ion Channel Genealogy. Ted mentioned the INCF, the Neuroinformatics Coordinating Facility. There's, of course, the Organization of Computational Neuroscience, which just had its annual conference a couple weeks ago. And we also make some announcements on the NEURON Simulator Twitter feed as well. So Twitter is a great way to stay up with scientific developments and what's new. With that, I want to, well, I'll pause right now and ask if anybody has any questions on what we talked about.
You discussed placing a model online, excuse me, as part of the review process. What about if someone wants to put up a preprint? Should they think about getting their model on ModelDB when they do the preprint?
We encourage you to do that. So that we encourage you to do that with an asterisk. So again, ModelDB's policy is that in order to be public, it has to have a associated peer-reviewed publication. So, well, what do you do with the preprint then? What you can do is, one way of doing that is you can go ahead and put the link to ModelDB in your preprint, because if you do go ahead and publish it, you know, you might as well have one link, and you might as well figure it out early, and you might as well have that in there, so that by the time it's published, it's there. But okay, well then that sounds like it's annoying to the people that are visiting it. Well, that's no problem. All these archive, like archive, bioarchive, they all have a comment section. So that would be one way. You could put in the comments that there's a read access password, you know, look, you know, here's this, and by the way, you need this password to get in there. Is it ideal? No, but it does at least get your model where it needs to be for the final publication. And it does give people access to it and the structured data. I don't know. Does anybody have any other suggestions for working with preprints in ModelDB? So that's, and incidentally, if you have strong feelings about the preprint thing, it is a perennial discussion, because I think at some point there's enough things that go on these on the archive and other related resources that maybe we're going to have to expand to include preprints. I'm not sure. Actually, so Ben, do you have any suggestions on how to address? I mean, so the reason that we don't do this is, you know, peer review offers a sort of a statement of quality to some extent. Do you have any thoughts as to how we avoid any issues there?
Unfortunately, no. It's actually, I was only asking because I wanted to think about managing the priorities of setting up code that someone else can use and getting the preprint ready. It's literally just a scheduling question in my case.
No, okay. I mean, what you could, I mean, a option is you could go ahead. So one of the things when I start a project, I go ahead and claim a URL on ModelDB. I go ahead and upload a small thing, and then I go back and upload a bigger one. And then that way I know the URL and I can go ahead. My paper draft, I've already got a line. You know, I immediately get a sentence in my paper. Model code is available at URL. You do that, you know, and a few hundred more sentences and you've got a paper. Any other questions or comments on this in general?
Okay, just to make clear that even if you set up a private model, even if you create a model entry, it will be private until you tell us that you want it to be released, and we won't make it public until we hear from you and see that it has been accepted for publication. That is the current state of affairs, and that will be, if we do change the policy with regard to acceptance by formal acceptance by a journal, that'll be something that we'll make very obvious if and when that ever happens. So it may be that we may have a second category of model entries that are public, and that second category may be those that have been released, not yet officially accepted for publication, but you know, somehow accessible to you at least, because there is a big backlog of review, and the review process can be, some papers linger in a not officially published state for a long time and yet have solid work in them. So we don't want to corrupt the scientific literature. On the other hand, we don't want to be a needless bottleneck either. So.
Well said. And I'm trying to see real quick if there is a, okay. So this one is a blank entry, reasons unknown. But so once you go in there, if you have a model entry, you go ahead and enter something, and then you go and you view your model entry. You can go back, you can hit edit, you can add new things. So I can start adding hippocampus CA1 pyramidal cell. I can update it, and so forth. I can go back and edit my own entries. up until the time, and only up until the time, that the model has been made public. You can go and upload new zip files if things change. And then when you're ready to release control and you've decided that this is what you want to be the version of reference, you can hit this request to make public, and that will that'll send an email basically to you acknowledging it as the person who entered it, and to the curator to go back and take a look at it to make sure that the public, that there's a publication, that everything's associated correctly, ideally that it runs, and so forth. So this is what it looks like from the inside. If you do discover so ideally, once something is made public, it doesn't change. There are occasional instances where things need to change for one reason or another. If that happens, we do have a system for it where we keep track of the history of the file. And the way to do this is just to email the curator. And I think it says,
Can we delete it once it goes public?
not directly. Again, that's a question for emailing the curator.
Okay.
We had one case once that I can remember wherein a model was uploaded and made public, and one of the faculty involved had no idea that this was happening, and it included code that they were not ready to make public. And so what ended up happening there in that case was they contacted us, and a version of the model was made. So their group made a version of the model public that just sort of assumed that provided the results of that other code, but not the code itself. That was a somewhat unfortunate situation, but I think that made everybody happy, and that was how that worked out. But usually, I mean, the idea for this is trying to provide an archival thing so that sometime 10 years from now, somebody can go and look at this and say, I want to know what they did in that paper. So changing and deleting are things we want to avoid. Any other questions or comments? All right, so 2 things left real fast. I want to actually run something because I want to show you how that works. Just to make sure that you've seen it, and then I want to talk about the exercises, which are basically a look at 2 ModelDB entries. So let's say I found a ModelDB entry that I'm interested in. This one will do. I can hit download zip file to download the zip file. And then it's going to show up in my downloads folder. So it's going to show up in my downloads folder. Blah, blah, blah. It's on the other. There it is. It's in my downloads folder. And here's all the files. Thank you. So here's the files from that model. Cool. There's a lot of model files. So let's actually try to run something. So I'll launch my terminal. I can change into my downloads folder and then CA1A beta. I can see from right here that, hey, look, all of my mod files are in this folder. At least I think all my mod files are in this folder. It could be somewhere else as well. If I wanted to check, it's 2 places to look, obvious places to look. I'll check the README. which is being weird. There we go. I'll check the README, and it'll say, okay, in the expanded archives folder, compile the mod files using the command NEURON model. Okay, let's do that. So that's one place. Just look at the README. How does it say to run it? And for one of your homework exercises, it has something that might surprise you about running it. So yeah, so you should always check the README. The other place to look is to go ahead and pull up the Model Views tab, look at one of these things, and then check under References, Run Protocol, how do I compile it? Change into the folder and just run NEURON IV model. Well, that's easy. And Grumpy Clang notwithstanding, this should work on any platform, Windows, Linux, Mac. You can always type in an IV modal. It goes, it compiles. It's compiling all of the mod files, and boom, we're done. So this says, well, how do I actually run figure 3 here? So these are instructions for running figure 3. It says enter in GUI -Python. This is almost always equivalent, but not 100% equivalent to just typing Python. So this is launching, it makes a difference as to who is the main process, if you know what a process is. Is NEURON running the show, or is Python running the show? Like I said, usually it doesn't matter, but sometimes it does. Since I'm following instructions right now, I'm gonna just do what it says, NEURON GUI -Python, although I happen to know that I could just type Python here as well. It does pop up. Wait, no, I lost it. But, well, anyways, it popped up the NEURON main menu, but I can say from NEURON import HGUI, because that's what it says to do. h.load file, mosinet.hope. And everything's popping up on my other screen and then disappearing, so that's inconvenient. Where's my, where it is? Let me try this again one more time. And for giggles, I'm just going to run this from Python. NEURON import h.GUI. You know what? I'm going to share my other screen because I think that's going to make this easier. Let's switch monitors here for a second. Okay, so you should see this. So there we go. From NEURON import h.GUI, h.load file. The instruction said to load mosinet.hope. You may wonder about that. It's a strange sounding name. It's really historical reasons for this, but a lot of ModelDB entries are going to have mosinet.hope or mosinet.py as their entry point. This is by no means required, but this was the instructions. And so now when I do this, it gives us this interface as we've seen before. I'm going to, and then I could go ahead and press the buttons, or we saw in the run protocol at the bottom how to actually run one of these things programmatically. And actually, since your homework's going to ask you to do that, let me just plop that up again real quick. I could press the button, but instead, I'm going to follow the instructions at the bottom of the run protocol. Said references, run protocol. How do I run this? I'm going to say h.fig3, open close parentheses, and then h.run. Okay. So h.fig3, open close parentheses, sets up for figure 3, and then h.run ran a simulation. We saw it go up and then go back down. And I imagine, if I had to guess, yeah, there we go. We set up for figure 5. We're running for figure 5 now. And so forth. So we can take a model, we can look at the instructions on ModelDB when they exist, hopefully, and go ahead and try to run a simulation. We can look, and this is the other thing that I want to make sure that you remember exists. We can go Tools, Model View, And so this is NEURON's version of the ModelDB, the model view tool that we saw in ModelDB as well. And so it pops up a cell. I can look at the 18 insert mechanisms. We saw before that KAD had varied with distance. And so here is, in this particular one, yeah, okay. At this particular moment here, we're looking at the obliques changing in a different way than in the one that we saw before. And that has to do with this being a, that has to do with details of how this is as a Alzheimer's model. This particular cell is looking at blocked A currents. And so that's why the distribution of A-type conductances has changed. But so in any case, you can go, you can open up model view, you can do the usual sets of analyses to get a graphical sense of what's going on in the cell. You can see, you can look down here. Hey, look, there's one eye clamp. It has a delay of 0, duration of 0.2, and amplitude of 0.1. So that's probably, there's no other stimuli mentioned. There's no references to a net stem or anything like that. So what we're probably looking at is there's just one cell that's getting one input. That's what's causing it to fire. It's that current clamp. Well, so if I were to go back here, there's one current clamp. So that's an H.I clamp. And so the 0th current clamp does not exist. But the 1st current clamp does. So there's a current clamp. I know its identifier. And then I know that its delay is currently 0. We saw, what did we see before? In the model, nope, that's the wrong thing, sorry. The current clamp has a delay of 0, a duration of 0.2, and an amplitude of 0.1. So we could look into this if I wanted to change the delay to be, if I wanted to change this to start at time 10, it's not going to update in the model view. I'm going to have to close it and then reopen that. So tools, model view. But now when I go in and I look at the current clamp, I'll see the delay is now 10. So you could imagine, and this is what I'm going to ask you to do in your homework, you could imagine that you find a current clamp is driving one of these cells. And by the way, here's where that current clamp is. It's at the far end of the axon for some reason. There's a current clamp that's driving one of these cells, and it's what's causing it to fire. But you want to run your own stimulus. You want to feed in a Poisson process. You want to do whatever. Well, just turn that current clamp's amplitude down to 0. Make sure that you get the cell at rest, and then go and add in your net stem. Right? So that's how you can take a thing, turn off the stimulus, and feed it your own stimulus. Or you could go back in and make changes. Does that make sense on the high level anyways? So then what I'm going to do, because I accidentally quit my, when I was having, come on, open quickly. When I was looking for, trying to find the lost NEURON windows, I accidentally closed PowerPoint. So I'm going to open that real quick because I want to talk about the slides. Sorry, talk about the homework exercises that we'll talk about on Tuesday. I'll just go ahead and slide. Cool. So for Tuesday, here's my my challenge for you, and we'll do these on, like I said, on Tuesday. I'd like you to look at 2 models. One of them actually involves a language known as HOKE. Doesn't matter, you can run it from NEURON just the same. So this is ModelDB entry 2488, and we'll actually talk more about that on Thursday, I think. But so look at this model, and I want you to look at, well, what are some high-level questions that you should think about? Well, who's the authors? Who made this model? And what question were they studying? What were they trying to answer with this? If you're using ModelDB's model view, well, how many different sort of model structures did they look at? And in particular, there's one model structure that's designated as 1C, the layer 3 pyramidal cell. How many sections does it have? How many segments? How do you run this model? What is it that's causing the cell to spike? And is this model temperature dependent? All right. So if you look at this, go ahead and run case 1C. Make sure that you can get it to run. See what happens. And then what I'd like you to do is I'd like you to turn off that which is causing the cell to spike and add your own stimulus. So a netstim with Poisson, that's a firing according to a Poisson process. So netstim.noise equals true. And use Python to record and plot the soma's membrane potential as a function of time. So take a thing, figure out what it's supposed to do, modify, turn off its stimulus, give it your own stimulus, and plot and see what happens. And so you can do this without it even being written in Python, right? That's the power of NEURON. But here's a model that is written in Python. So this is ModelDB entry 239421. Start out some of the same questions, right? Who are the authors? What questions were they studying? What ion channels? But so for this one, try and figure out where the parameters are specified. So try to look through the code, see what kind of sense you can make of it. Run the model. I'm going to point to you that there are some special instructions in the README for compiling and running. I was a little bit surprised that my naive solution didn't work. When you do run it, it might be fun to look at your task manager at its top in Linux and Mac and see the CPU usage. So this is a model that will automatically parallelize. This is using thread-based parallelism instead of MPI, which is what we focused on. But NEURON also supports using threads to parallelize things. Some of the same questions, though. How many sections in the model? How many segments? Try and do this without using model view. There's a somatic leak conductance. If you were to double the somatic leak conductance and make no other changes, you'll notice a qualitative change in the model's behavior. Okay, so there's the baseline, and then there's twice as much leak conductance, and then that is a qualitatively different behavior. See if you can figure out where that behavior shifts. Is it just at the double that it shifts? Is it somewhere in between? What's going on? And I'll put all of these slides on the Slack, and these are the last two slides of today. But does that make sense? So I want you to look at these two models. They're semi-random. One of them is more random than others. But take a look at them and see if you can get them to run, see if you can make sense of it, see if you can change something. Any questions on that or anything else? Well, if not, I hope everybody has a wonderful weekend, and I'll see you on Tuesday.